The overall goal of this imaging procedure is to standardize bioluminescence acquisition procedures and image analyses to obtain quantitative data for comparison within or among different experiments.
This method can help answer a key question in the bioluminescence imaging field, such as how can imaging data be quantified in a reproducible way. The main advantages of this technique are that it is easy to set up in any animal facility that owns a bioluminescence imager, and that it does not require complex calculations.
Turn on the imager and open the acquisition software. Click Initialize to initialize the camera, stages, and lenses. After confirming the appropriate level of sedation by lack of response to leg pinch, place a mouse on a warming pad and apply eye ointment. Then, use a 25-gauge needle to inject 100 microliters of freshly prepared D-luciferin potassium salt solution intraperitoneally. In this case, tumor cells express firefly luciferase. This protein oxidizes D-luciferin and produces light that can be detected with the imager. After 10 minutes, place the mouse prone into the imager with its back facing the camera in as flat a position as possible. Select the auto exposure feature and click Acquire to obtain